Hello, my friends. In this video, we'll be taking a look at the autosomal DNA results, predicted phenotype traits, and GED match, and even G25 results of the of an inhabitant of Suzdal, which is a city in central Russia, a couple kilometers, maybe 200 kilometers to the east of Moscow in Vladimirskaya Oblast, the city of Suzdal in central Russia. The sample name is GOR001, and this individual lived in the 8th to 10th century AD in Suzdal in Vladimirskaya Oblast. Now, Suzdal region is the historical core of northwestern Russia. It says northwestern Russia here on illustrative DNA, but I completely disagree. I don't think Suzdal is northwestern Russia at all. I think it is the definition of central Russia and has nothing to do with north or west. Like, why do they say it's northwestern Russia? So I disagree with them saying it's northwestern Russia. In the 11th to 13th centuries, it is characterized by a continuous existence of rural settlements that emerged in the 10th to 12th centuries, often in territories peopled by, in the first millennium Common Era. The region is considered part of the area inhabited by Uralic-speaking groups until 9th to 10th centuries, while the Slavic colonization began in 10th century AD. So in 10th century, the Slavs come in, and the Uralic people sort of get pushed out of Suzdal. And that's what we see with samples such as GOR001, which we're going to see. So this person is part of the early Slavs that come in and start pushing out the Uralic people who used to live in this region before. It's one of the early Slavic people who actually come into this region. Medieval cities Suzdal, Vladimir, Yuryev, and rural settlements in Suzdal region have been thoroughly studied by archaeologists. The material culture of these is found to represent classical Ancient Rus tradition of the 11th to 13th centuries. The preceding period is characterized by a complex and multi-component material culture combining elements of both Slavic and Volga Finnic groups. So what's interesting about this early medieval Suzdal, for example, is that you can see the closest modern groups to it are Russians from Pinega, Krasnoborsk, Karelians, Mordva, Finns, and various northern Russians. So they are quite distant from central Russians. And I'm actually pulling this up from my closest groups on illustrative DNA because I can't. There is no other place I could generate. A report like this where it actually gets you all the information about this sample. It's really cool that they show you all the information. I like the information part. The closest group to me is actually post-medieval Russian from Suzdal, which is very, very different. If you look at the distances to modern population, you can see that this is actually like a modern, typical a human, like normal average central Russian. It's close to Tver, Ryazan, Kursk, Oryol, Yaroslavl, Voronezh, and then Ukrainians from Dnipro and Donetsk. So like Russian from post-medieval Russian from Suzdal, it's already like a typical Slav. There is almost no Uralic admixture whatsoever. But then if you look at early medieval Suzdal, there is much less Slavic admixture, and it's much more Finnish, much more Uralic. So what we're going to look at the results of one of those early medieval Suzdal people. We're going to look at the results of one of those people who are from the early medieval period from Suzdal, who are much more Uralic, who have less Slavic admixture. We're going to look at GOR001. We're going to look at this sample. In case you don't know where Suzdal is located, let me show you where that is on the map. So, obviously, you cannot say that this is northwestern Russia. This right here is not northwestern Russia. It's not even close to northwest of Russia. It's very central Russia. It's not really even north of Moscow Oblast. If you draw a straight line from this to here, it falls into Moscow Oblast. If you draw a straight line from this to here, it does not fall into Belarus, but it falls into Lithuania still. So it's not northern Russia whatsoever. It's not northwest Russia. Okay, it's not that northern in terms of where it's located. But anyway, yes, this is Suzdal. This is where Suzdal is located. It's a little bit east of Moscow. Let's see how many kilometers is that east of Moscow. Let's measure. That's looks like it's 118 miles to the east of Moscow. So in kilometers, that would be maybe 200 kilometers to the east of Moscow. Quite distant from Moscow. to the north and east. So in Russia there's this concept of historical Russian cities. It's called the Golden Ring. I think it includes cities like Kostroma, Suzdal, Vladimir. I know it includes Murom. It includes Yaroslavl. It includes Rostov. It includes Galich. It includes, I'm not sure where Galich is. It includes Uglич. I know it includes Pereyaslav, Zalesky. Um, I'm not. It includes a bunch of cities like that. It includes Moscow for sure, and it includes Ryazan, I think, as well. So this concept of Golden Ring in Russia, um, it's like stereotypical like core Russian cities of historical significance, right? So Suzdal is right in the center of that. So it's not northwestern Russia. It's not some outskirts city in the north of Russia. It is historical core of Russian development and Russian ethnogenesis. It is as central Russian as it gets, at least in my opinion. Anyway, let's move on to the results of this individual. First, we're going to start with the G25. I simulated their G25 using raw DNA to simulate the G25 tool with the tool that's on Explore Your DNA. So this is what I'm getting with the Davitsky model for G25. We're getting 56% Yamne, very little Barsen Neolithic. We're getting a little bit of Nanganasan and Han, so a little bit of Uralic admixture, as we could expect. So this person does have a little bit of Uralic admixture that we can see with G25 as well. And the closest modern populations with G25 seem to be Erzya, Russians from Kostroma, Russians from Krasnoborsk, Moksha. Followed by that are Vepsians, Russians from Pinega, Pineshki. So Pinega and Pineshki are northern Russians. They are from the very far north of Arkhangelskaya Oblast. So that's obviously much more north than Suzdal is in terms of location. Karelians are also very to the far north. Ingrians, various Finnish groups are also very close here. Then come Russians from Yaroslavl, Lishukonsk, North Finns. But overall, it looks like this person is actually a little bit closer to Central Russians, such as Kostroma and various other groups that are close to Central Russians, like Erzya and Moksha, than they are to Northern Russians, such as Pinega or Pineshki. Because as we can see here, for early medieval Suzdal people, they are closer to Pineshki and Krasnoborsk than they are to Mordvins from Erzya or Mokshas. So this early medieval individual from Suzdal, GOR001, actually seems to be a little bit more southern and a little bit more Slavic than the rest of the samples in this than the SHE003, SHE004, and SHE006 samples. Let's see what they score with the single mode oracle. They seem to score a mixture of 39% Erzya, 33% Kostroma, 23.8% Lishukonsk, and 3.6% Pineshki. All of these groups are either fully Uralic or partially Uralic. Kostroma would be the least Uralic group out of all of these. Kostroma is the most Slavic group out of all of these four. Erzya is the second most Slavic group out of these four. Kostroma is, you could say, is half Slavic, half Uralic, and Erzya, you could say, is slightly more Uralic than Slavic, and Lishukonsk is a lot more Uralic than Slavic. But and Pineshk is also a lot more Uralic than Slavic. But Pineshk is kind of weird because it also has like a very Paleo-European component. But let's try to reduce it to three populations. Let's see what we get there. So if we reduce it to three populations, we get a model like this, where it's 58.4% Lithuanian, RA, 34% Udmurt, 7.6% Slovakian. Let's remove the Lithuanians. Lithuanians are very northern and they confuse the result. Let's remove the Lithuanians from here. They're kind of irrelevant to our quest to find out their true ancestry. So now that we remove Lithuanians, we get a result like this, where it's 36% Russian Ryazan, 34.4% Latvian, and 29% Udmurt. So definitely very, very Uralic admixture, a lot of Uralic admixture. So a lot of northern and eastern admixture. Scoring so much Latvian relative to Russians from Ryazan is a sign of excessive northern admixture, and scoring so much udmurt is in the sign of excessive eastern admixture. So this person is certainly very Finno-Ugric. So relative to Russians, relative to Russians from Ryazan, this person is excessively northern and eastern. Still, though, it looks like he is more Slavic than these other samples like 6003, 6004, and 6006. Let's see what this individual scores with Yerujinsky 13. Here he's scoring 51% Baltic, 22% North Atlantic. He's scoring 7.5% Siberian, which is very much. It's a huge amount of Siberian to score with this calculator for anybody from Eastern Europe. He's scoring very little West Mediterranean, only 5.7% West Mediterranean. But aside from that, his score for West Asian and East Mediterranean is actually quite in line for Eastern Europeans. So when it comes to Mediterranean components, if we ignore the very low score for West Mediterranean, he's actually scoring quite in line for what's typical for Central Russians. So because of that, he's scoring closest to Erzya and Kargopol Russians. Kargopol is in Arkhangelskaya Oblast. So once again, he's not all that distant from various Russian groups. So he's not really all that Finnish, not all that Uralic. What's also interesting is he's scoring very little East Asian. So he's scoring a lot of Siberian, but not a lot of East Asian. 2.29% Amerindian. If we add up the Amerindian and the Siberian together, that's gonna be 9 point, I think 9.80 something percent East Eurasian admixture in total. He's scoring 2% South Asian, which is so much South Asian admixture. That's crazy. Okay, that's quite interesting. And for the four-way population approximation, looks like the closest four-way to him is a mixture of 25% Erzya, 25 half Lithuanian, and 25% Mari. So the closest four-way is quarter Erzya, half Lithuanian, and quarter Mari. That's very, very interesting. Not even a bit Slavic. Very interesting four-way. Okay. Now let's see what he scores for my own ethnicity calculator. This is definitely going to be very interesting to see. With 415 SNPs, he is scoring closest to Jewish Ashkenazi, followed by North Germanic, followed by Azerbaijan Neolithic, followed by French. Okay, that's completely out of the left field. Very wrong. But you cannot really get a very precise prediction with so few SNPs. So in some cases it's gonna be quite good, but in other cases, like in this case, for example, it is completely wrong. And the two-way models is a mixture of Vez 56 Tollens Germany Bronze Age plus British Neolithic farmer, Vez 56 Tollens Germany Bronze Age plus Kafkas medieval individual from Anapa, Vez 56 Tollens Germany Bronze Age plus Velikent, Vez 56 plus Sarmatian, Vez 56 plus Turkish 2. So it looks like the two-way model seems to be modeling him as a mixture of Vez 56 Tollens Germany plus a mixture of something else. That's quite interesting, but it's obviously quite wrong. And I'm noticing that the distances are quite large to everything, to Jewish, to North Germanic, to everything that follows it. The distances are quite significant. But it is quite a bad result. Quite a bad result. Okay, let's close that. And now let's see what he scores for Nashakot. Let's see what he scores for Phenotype and what he looks like and everything else. So for Phenotype, it looks like his predicted eye color is blue. Looks like he has blue eye color. Blue eyes with an Ebersender ring are also quite possible at 38.7% likelihood. So he most likely has blue eyes or blue eyes with an Ebersender ring. Green eyes are kind of possible at 8%, but not really all that probable. Hazel eyes are not very probable at 6%. And brown eyes or darkest brown eyes are completely improbable. Very, very improbable. For hair color likelihood distribution, he most likely has dark blonde hair or red hair. Either one is possible for him. Dark blonde or red hair. Light blonde hair is not very possible. Light brown hair is possible but not very probable, and dark brown hair is not very possible at all. Black hair is not possible at all. So most likely this person has dark blonde hair or red hair. And for skin color likelihood distribution, looks like this person has white skin tone. Actually, white or palest skin tone. Very interesting. For hair texture, looks like he has curly hair, although kinky and wavy hair is also quite possible. Uh, Definitely, it goes against the stereotype that Russians have straight hair. But I have seen quite a significant number of Russians with curly hair texture. So, including people in my own family. So curly hair texture is interesting. Curly hair. And for nose shape, looks like he has a Greek shape nose. That's the predicted nose shape for him. So blue eyes, blonde, dark blonde or red hair color, white skin, and curly hair, and Greek shape nose. Okay, now let's see for coloring related variants. Looks like he has heterozygous genotype for blue eye haplotype 3. Looks like he has blue eye haplotype 2. Looks like he probably has blue eye haplotype 1 as well. We don't know because he doesn't have a genotype here, but we can predict that he probably does as well. Looks like he has 2 light color variants in this variation of SLC 24A5 that has to do with color of light hair of eyes, hair, and skin. 2 light color variants in this variation of SLC 45A2 that have to do with the same thing. Okay. And he actually has 2 light color variants in these variations of MC1R that have to do with the light pigmentation of hair. Let's see what he scores for hair with snipper free. I'm curious to see if they will also classify him as red haired. Let's see. I think not, because they don't have such a wide range of markers they look for MC1R. But I will find out. Oh, they do. They actually also classify him as red-haired. That's interesting. So they classify him as 2.63 times more likely red than blonde, and 306 times more likely red than brown. So Snipper Free actually also classifies him as red-haired. That's definitely quite interesting. So the Snipper Free also classifies him as red-haired. Definitely quite cool. Let's see the phenotype oracle. What does that look like for him? For him, the phenotype oracle looks like this. The closest phenotypes to him are the following. And this is the closest phenotype to him, followed by this, followed by this. These are all the closest phenotypes to him in terms of what they look like. Okay, so this, followed by this, followed by this. This is like a Nordic, Northern European phenotype. This is like a Finnish, Finno-Ugric, but also like sort of Northern European as well. And this is like once again Northern European phenotype, but sort of more potato, a little bit overweight looking. And for the mixtures, he seems to be getting more or less a mixture of 50% this plus 50% that. 50% this plus 50% that for the second closest mixture. In case you aren't getting the little differences, the subtle differences is that here on the bottom the person has red tint in their hair color. The third closest mixture is 50% that plus 50% this. The fourth is 50% this plus 50% this. So very interesting models for sure. Okay, let's close this. And what do they score for the biomarkers panel? Looks like they have a predisposition to lower than average levels of vitamin D, higher than average levels of LDL cholesterol, below average levels of HDL cholesterol, below average levels of glucose, above average levels of hemoglobin, above average levels of blood pressure, below average levels of iron in the blood, so probably don't have hemochromatosis, above average levels of sex hormone binding globulin, below average levels of red blood cell count in blood, above average number of base pairs, so it looks like they have predisposition to longer telomere length, quite good, and below average height. Okay, quite interesting. So they have a predisposition to longer telomere length. That's quite cool. Let's see what they score for the polygenic risk scores. So looks like they have a much below average odds for hemoglobin E disease and no risk variance anywhere. Looks like based on 8 S&Ps, they have a below average odds of migraine. Looks like they have a above average odds for lupus, very high odds for lupus. Looks like they have very low odds for gout. Looks like they have below average odds for thyroid cancer. Looks like they have pretty much average odds for eczema. Looks like they have slightly below average odds for exfoliation glaucoma, average odds for pigmentary glaucoma, average odds for open angle glaucoma, and average odds for primary congenital glaucoma. Looks like they have average odds for polycystic ovary syndrome. Looks like they have very high odds for cataracts. Looks like they have slightly above average odds for age-related macular degeneration. Looks like they have average odds for rheumatoid arthritis. Looks like they have average odds for Tourette's. Nothing was found for epilepsy. Looks like they have average odds for asthma. Looks like they have above average odds for leukemia. Looks like they have average odds for vitiligo. Looks like they have below average odds for myopia. Looks like they have below average odds for corneal astigmatism. Looks like they have average odds for primary biliary cirrhosis. Looks like they have average odds for stroke. Looks like they have below average odds for male pattern hair loss. Looks like they have average odds for atrial fibrillation. Looks like they have nothing relevant was found for deep vein thrombosis. Looks like they have, based on 9 S&Ps, slightly above average odds for unipolar depression. Looks like based on 25 S&Ps, they have very high odds for bipolar type 1. And based on 22 S&Ps, they have very high odds for schizophrenia. Very interesting. So they definitely have a predisposition to bipolar type 1 and schizophrenia based on the stuff that's in the file. Looks like they have a average score for type 2 diabetes and a very high score for type 1 diabetes. Looks like they have slightly below average odds for Alzheimer's. Looks like they have slightly below average odds for multiple sclerosis. Looks like they have 2 risk variance for breast cancer that have 12, which is kind of typical, and 11 risk variance for testicular cancer of 20. Two risk variance and all the important variations of kilo G, which is kind of unfortunate. So they have all the risk variance and all the important variations on kilo G. That's kind of unfortunate. Celiac disease, 3 risk variance out of 8. Once again, kind of unfortunate. No risk variance for GSS, 7 out of 22 for Crohn's, but none of them are in the important variations. No risk variance for Riefenstein's out of 4. No risk variance for Parkinson's out of 12. 3 for Gilbert syndrome out of 4, but these are typical, I think. No risk variance for basal cell carcinoma. No risk variance for GCD. No risk variance for Vardenberg. No risk variance, 2 risk variance for IBS, but they are common. 2 risk variance for Brugada, but they are common. And all the risk variance they have for all the following syndromes are all common. So, okay, what we saw here that was very surprising is the risk score for schizophrenia, which is very, very weird. 4.13 times the average, and for bipolar type 1, which is 3.42 times the average, and that is very, very high and is very unusual. That's definitely quite an unusual score. Okay, so that's the highest thing they score for, actually. That's the highest, these conditions are the highest scores that they scored for. So let's explore the results and find out why they scored the way they did. For results for mental health section, looks like they have predisposition to warrior phenotype, slower dopamine reuptake, and higher dopamine levels, lower stress resiliency. Looks like they have AA and Combs-Met variation, which means warrior genotype and Combs-Met. So slightly less of the COMT enzyme, and therefore slightly slower breakdown of dopamine, more dopamine in the system. Also, warrior genotype in this variation of COMT. Okay, also, do we have any other warrior or warrior genotypes? Not really. I don't see anything that would particularly say warrior or warrior here, although they all contribute to something. I don't. I should have explicitly written warrior or warrior in the alleles. Okay, for D2, looks like they have a predisposition to higher number of dopamine D2 receptors, so that definitely contributed to the score for schizophrenia and bipolar. They don't have any derived no goal evidence and DRD2 sporophrines in pro variation, which means a higher number of dopamine D2 receptor sites in the brain and higher number of higher likelihood of schizophrenia. This variation is also linked to the no-go learner variation. So they typically you inherit the A allele here together with the G allele here. So once again, here the AA genotype here comes together with the GG genotype as well, which means once again higher number of dopamine D2 receptor sites in the brain and higher likelihood of schizophrenia. CC in this variation of DRD2, which also comes together with a higher odds of schizophrenia. AA genotype in this variation of DRD2, which is implicated in a higher number of dopamine D2 receptor sites in the brain and higher risk of schizophrenia. Once again, this is a very uncommon genotype to have. Actually, it's a very, very uncommon genotype, and I'm surprised that this genome has it. So this definitely played a big part in the polygenic risk score. This is a very interesting genotype. If you look at the genotype distribution for this genotype, it's very uncommon to have AA. Like for Europeans, it's so uncommon. And if you do some research, the frequency of the A allele in people with schizophrenia is much, much higher than the G allele. Okay, for TAC1, this person does not have the A1 allele in TAC1. Once again, so higher number of dopamine D2 receptor sites in the brain. So this also contributes to a higher risk of schizophrenia. And another genotype here that also contributes to a higher number of dopamine D2 receptor sites in the brain. Another genotype that contributes to higher number of dopamine D2 receptor sites in the brain. So overall, we can see why this person scored the way they did for the risk of schizophrenia and bipolar. It makes sense. They have this genotype, but does not play a role in polygenic risk score calculations, so this also makes sense. Okay, they have heterozygous genotype in ADCY2, which is associated with bipolar disorder. They have this genotype in CRANK1, which is also associated with higher odds of bipolar and schizophrenia. This also plays a role in calculation of the risk score. For the 5-HT and serotonin panel, they have a predisposition to intermediate or lower levels of 5-HT and serotonin. So it looks like they don't have long-form 5-HTT LPR. They do not have a protection from depression and anxiety. For FGFR3 gene panel, looks like they do not have dwarfism, which is really good. For insomnia and sleep disorders panel, looks like they have an uncommon genotype here, which leads to a 7 times increased risk of DSPS, according to a 2001 study. Very, very interesting. Okay, for lactose persistence, this individual is actually homozygous for European lactose persistence mutation and is almost definitely not lactose intolerant. Very good to see. So this is one of the few cases where we see a person actually having homozygous genotype for the European lactose persistence mutation. Very cool. For OXTR and empathy gene, this person has a predisposition to lower levels of empathy. Based on these 4 genotypes that were found in the file, definitely a predisposition to lower level of empathy. For diabetes, remember they were scoring, I think, low for type 2 and high for type 1. Let's see again. Yeah, they're scoring average for type 2 and high for type 1. So we see we're not going to look at that actually. For hemochromatosis, looks like they have looks like they don't have any risk variance for hemochromatosis. For Alzheimer's, no genotypes for APOE, and actually no, they do have a genotype for APOE. No risk variance in APOE. For multiple sclerosis, no risk variance in HLA, which is really good to see. For cardiovascular gene panel, cardiovascular disease panel, looks like they have a predisposition to the lower odds of cardiovascular issues, which is really good to see. So they don't have any predisposition to cardiovascular issues. Really, really good stuff. Okay. For facial morphology panel, looks like they do not have any East Asian genotypes in EDAR. Fully European genotypes in EDAR. They have a genotype for shorter mid-face length, higher odds of protruding nasal bridge, intermediate nose size, and a genotype for slightly thinner eyebrows. Very interesting. For vitiligo panel, they have a genotype. They actually have a risk variant for vitiligo in the HLA gene. Definitely quite cool. For miscellaneous section, no micropenis. They have one fat gene variant in FTOs RS9939609, which leads to high rates of obesity and sleep apnea. They do not have the Asian flusher mutation in RS671. And for alcoholism panel, they have slightly below average odds of alcoholism. For drug response, looks like their risk of heart failure due to beta blocker medications is slightly below average. Okay, they have this gene type in OCT1, which leads to lower odds of cannabis-induced psychosis. They are not carrier of a cutaneous albinism type 1b mutation. They do not have any risk variants for familiar Mediterranean fever. For MTHFR panel, they do not have. They actually have a genotype here that leads to possibly impaired folate metabolism in MTHFR, which is kind of unfortunate. For leukemia, they definitely have a lot of risk variants for increased risk of leukemia, and they were scoring high for the leukemia panel again. For rare diseases and traits panel, let's see, do they have any predisposition to any rare conditions? They have a predisposition to tuberculosis infection if exposed to tuberculosis bacterium. So they actually have a predisposition to, if they are exposed to tuberculosis bacterium, they are much more likely to contract tuberculosis, which is definitely quite interesting. They have a risk variant for leprosy, and looks like that is it. They have a protection from male pattern hair loss in the AR gene. In this variation of the AR gene, they have AA genotype, which protects them from going bald. And they do not have any protective variants from HIV in this variation right here. For the Canavan syndrome panel, they do not have any risk variants for Canavan syndrome. For the HLA gene panel, looks like they have a predisposition to intermediate or slightly higher odds of autoimmune disease. So they actually have a predisposition to slightly higher odds of autoimmune disease based on the genotypes in HLA. Kind of unfortunate. For MTRR gene panel, looks like they do not have any risk variants for neural tube defects or disorders of collagen metabolism, which is really good stuff. So really healthy. For homocysteine panel, looks like they have predisposition to average or intermediate levels of homocysteine, which is really good. And for muscular dystrophy myopathies, looks like they do not have any risk variants for any muscular dystrophy myopathies. Really good to see. For color blindness panel, no risk variants for color blindness. For FTO gene panel, looks like they have a genotype which homozygous for the fat gene. So definitely a predisposition to obesity. And another genotype which is heterozygous for the fat gene, so slight predisposition to obesity. And then they have some other genotypes which are not in the FTO gene, which correspond to lower BMI and lower predisposition to obesity. So if we only take into account the FTO gene, then there is definitely some predisposition to obesity. But if we take everything into account together, then really pretty much average predisposition to obesity. For syncope panel, based on 3 SNPs, this person definitely has an above average predisposition to syncope. And for bio traits panel, I'm going to ignore some of this. I don't want to discuss all of it. This person has 2 copies of the hunter-gatherer CLT-CL1 gene variant, which leads to a reduced ability to process carbs and sugars. This person is more able to detect beta-iodine floral fragrance. For vitamins and levels panel, this person typically has lower levels of vitamin K, but this person also has another genotype for higher levels of vitamin K, lower levels of vitamin A or retinol, and intermediate. That stuff's super boring. We're just moving on to the blood type. So the blood type for this person is most likely blood type A. Blood type A is 60% likelihood for him. Blood type O is 21% likelihood for him. Blood type B is 12% likelihood for him. Type AB is 5%. So most likely this person has blood type A. So probably has blood type A indeed. Although this was determined with only, let's see how many SNPs that is. That's 1, 2, 3, 4, 5, 6, 7 SNPs. So based on these 7 genotypes, most likely this person has blood type A. Well, thanks for watching until the very end. Leave a like and subscribe if you enjoyed it. I certainly enjoyed making this video. And also make sure you go to my itch store page, which is in the description of the video, and check out my tool that I use to generate all this content, that I use to generate all these reports, on the ancient genomes that I analyze. This will work on your raw data file, MyHeritage, 23andMe, whatever company you use to DNA test yourself with. It will work on your raw data file. And also, it is only $13 currently, but starting on May 22, it will be a lot more expensive. But also, the link to download the file, the data file that I've been analyzing in this video, it will also be in the description of the video as well. Thanks for watching until the very end. Leave a like and subscribe. Goodbye.